Okay, let's have a class, and we will first of all move to exam 3, talk about genetics and biochemistry, and also applied microbiology later on. I will talk briefly about the microbial genetics, and I also upload onto the eCampus, which is a case study for you to practice for the exam, which will be on Thursday. Thursday exam, still the same open book. We will have a 5 question to practice at the end of this class. Do not forget, very important is 3 file, which is for after class reading. Please do definitely go over it today, tomorrow, or at least before the exam. Then you know what it is. Okay, because some question you may need those to answer. So that's very important. Just want to talk to you before. So exam 3, we want to talk about the genetics and the biochemistry. And also applied microbiology will not be very detailed for genetics part, but some of the key points we would love to touch on. So when we talk about the microbial genetics, we always will go through central dogma, which is usually the outline of our class, which is encoding DNA, go to the RNA, and then go to the protein. And you learn in your different classes. So DNA, first of all, is doing replication. And then they will go to RNA. This is called transcription. And then finally will be expressed as a protein. So we call it is translation. That is outline, and but something else we wanted to say, with the new things could happen is this guy could go back. This is called a reverse transcription. That's also could be part of all the supplements for the central dogma. Okay? Therefore, our class will be talking about DNA replication, transcription, and translation. But compared to the other classes, will be relatively brief. Basically, the idea we will touch, the knowledge we will touch, but not going to go tedious detail. Okay? So today, we're going to talk about the DNA before we talk about the DNA replication. So we're going to talk about DNA. When we get to detail of the DNA, I would love to mention several very important studies in the world, in the history. So we want to go over the history a little bit. First of all, people start to do the research of DNA is actually 1912 Spanish flu. By the time the Spanish flu is as damaged or more damaged than COVID-19. Kills millions of people in the European countries. And the people wants to, the scientific area wants to develop a vaccine. So they started to do something related to microbial genetics. And by the time, they don't know what type of the material transferring genetic information. So they try to solve the problem is transformation principle. That is what they're trying to do. Therefore, there are a bunch of important research has been done in the history. So we want to go over these. First thing, in 1928, F. Griffiths did his very important mice study. So what happened? I have Streptococcus pneumoniae. And we already know Streptococcus pneumoniae cause number one community-acquired ammonia in the United States. There are two different types. One is smooth. Another one is rough. The smooth one, usually we call it virulent, which means it will cause ammonia for killing animals or human beings. The rough one is a surrogate, which means non-pathogenic, you can say, as a control. Now, when F. Griffith started to do this research, what he did is to did on the mice. He did a bunch of lots of the study using animal as a model, which using mice as a model. So what he did, I have a smooth Streptococcus pneumoniae. I'm going to inject it a mice. It's a virulent one. What happened? The mice is dead. Okay, the mice is dead. As a control group, I'm doing a rough one, a rough Streptococcus pneumoniae to inoculate it or inject it another mice. Obviously, because it's not virulent, the mice is alive. It still survived very well. Okay, I have another control group. I have a smooth Streptococcus pneumoniae. However, I heat it. So I do heat injured. And you know the bacteria, when you heat it, what's going to be? They're going to lose virulent function. So they inoculated it to a mice. The mice is live. Okay, let's do something special. I'm going to combine a rough Streptococcus pneumoniae with a heat-injured smooth Streptococcus pneumoniae. So this is a smooth Streptococcus pneumoniae. But I'm heat-injured. I'm also going to inoculate it a mice. What happened? The mice is dead. What tells you? Something happened right here. A rough surrogate Streptococcus pneumoniae and a smooth heat-injured Streptococcus pneumoniae combined together, the mice is dead. There's some communication here. Related to transferring genetic information. Today we know it is DNA. Today we know this method is called transformation. We know about it because we already did a lot of research. By the time we don't know. We only know there is some genetic material there. So this is a very important research in 1928 by F. Griffiths. His group later on did tons of animal study. Thousands of, hundreds of mice died every day and every year. And he made some progress later 1930s. Unfortunately, in 1941, F. Griffiths was killed by German bomb during Second World War. A very talented young scientist, not really young, a scientist was being killed during the Second World War. So people have to do something. People need to find what is this transformation information it is. Luckily, we have a big group to do. So where are they? This moves on to our next stage of our research. Done by in 1943. During the Second World War, everybody is busy, but the scientists are still working. In Rockefeller Institution in New York State, in New York City, right now is Rockefeller, by the time it's Rockefeller University, right now is Rockefeller Institution of Medical Center. There is 3 great American scientists. Avery, McCarty, and McLeod. They did a bunch of study. They're using RNAs, DNAs, and proteases using tube. Okay, why this is, I want to say it is important. This is did in a mice, using animal as a model, and it's pretty expensive. We call this is in viral study, is that right? This is in vitro study, which is make a big progress. And finally, he got isolated. They got isolated DNA. And they make sure, or proposed by the time, this genetic information, it is DNA. And the detailed research is there, and you can read. I'm not going to go through that one because my major class, AEM 341, we spend another 15 minutes talk about this. We don't, in here we'll just go briefly. Okay, Avery, McCarty, McLeod in 1943 get isolated DNA. So once you get isolated DNA, what are you going to do next? We're still going to have to do something to verify it. And we from different perspective. This is what happened. In 1952, another great scientist, Alfred Hershey Chase experiment. Okay, this is not a candy ball. Okay, Hershey, a chocolate, not a chocolate ball. It's Alfred Hershey. And this is not Chase Bank. It is Martha Chase. And they did a great work. They, from different perspective, what they do, they using a bacteriophage as a tool. What is a bacteriophage? A bacteriophage, which means a virus could either eat a bacteria, burst a bacteria, or they could either integrate their genome sequencing into the bacterial sequence. And the phage structure is very simple. The outside, we call it a capsid, which is basically is protein, and inside, it is genomics. It is either DNA or RNA, but never both. And luckily, when they choose a bacteriophage, which is a T4 phage, it is a DNA phage, and it is a temperate phage. We call it a warm phage or friendly phage. They could integrate their sequencing into a bacteria. So what they do? We wanted to see what happened. We need a label. So what they do? Bacteriophage is pretty simple. There's only protein. What chemicals is very special for protein? Sulfite, is that right? And I'm gonna using isotope to do the label, which is S35. That's isotope. What is special for DNA? What's a chemical very special for DNA? Besides CHO. We'll talk about DNA structure real quick. A phosphate. Is that correct? A phosphate. So he labeled the phosphates use 32. And then we're gonna find a bacteria. So we're gonna find the E. coli. Just to find a basic E. coli. Okay, there's a bacteria. The bacteria genome is like this. Okay, what happened? This guy gonna come in, and then they're gonna be integrated into the bacteria. Of course, they could have some new bacteriophage will be generated. Okay, and we know that they are using. Virus is an obligate parasite. They cannot replication, transcription, translation by themselves. They need a host. So how do they do it? During this research, there is a certain stage which is a mixture with bacteria and the phage. So what they do by the time? They don't have a centrifuge. Right now you can do high speed in centrifuge. Then you have a precipitation, you have a supernatants. By the time they don't have it, 90-50. What do they have? Those guys are really smart. They use kitchen blender. Like you make juice. Smoothie, just blend it. Use a kitchen blender. They got the isolates. What they got the isolates? Bacteria basically is at the bottom. Because it's relatively heavy, so bacteria is at the bottom. Virus, phage is at the top. And what did he find? He found that it's very interesting. In the bottom bacteria section, they only see P32. There is no S35. What this means? Which means the information transfer from a bacteriophage go to the bacteria. It is DNA. It is not protein. Okay? That's very interesting. I was telling you here is a short story. Okay, long story short. If you go Wikipedia or you do some textbook, it's more complicated than that. Just to tell you a short story. So this is 1952. Then time moves quick. We're going to talk about a big deal. Not enough space, so we're going to remove it. So we talk about 1928, F. Griffith study. 1943, every McCartney find isolated DNA, and in 1952, Hershey-Chase verified that from bacteriophage standpoint, no, it is DNA. Okay. So time goes very quick. 1953. There is two great scientists in the world. James Watson and Francisco Crick. Francisco Crick passed away, James Watson student. By the time James Watson is a newly PhD graduate student from Indiana University, Francisco Crick is a physician, worked for Great British Army. Right after World War II. And James Watson be a visiting scholar in Cavendish Lab in Cambridge. Then they meet with each other. They have like a 12 years old age difference, but they get along very well. They go there, they did do research. What they do, they talk, they have a cup of tea every day, cup of coffee, lunch, dinner, they just sit there and talk and thinking. What's the structure of DNA? It is very interesting by the time. Now, we call those are theoretical biochemists. Not happening anymore today. Today, we all need to rely on experiments. What happened after a couple of days? They are very lucky to meet a great British woman scientist, Rosalind Franklin. Rosalind Franklin showed them the research work she did using X-ray diffraction photographer and find the DNA very much possible. It is double helixy. Okay, now, how do we know the DNA structure is that? Not done yet. We have ATGC4 base. Okay, ATGC4 gene base. We know about that. How they connected with each other. They're not done yet. Because at the same time, a great American chemist, Linus Pauling, also find a similar structure. He also predicted the DNA structure could be double helix. And Pauling published a really small paper. But luckily, James Watson and Francisco Crick met another great scientist, a chemist from Columbia University, also visiting Cambridge University by the time. Shagraf. This gives you a very important, Shagraf had a lunch with them, tell him a very important information. In a certain biological species, you could have a different A and T, you could have a different G and C, but the ratio of A and T and G and C is always 1:1. This is bingo, comes out with double helix. And in 1953, they published one piece of paper at Nature. People say it is the most beautiful, the single most beautiful biological paper in the 20th century. Only one page. They talk about the DNA structure. It's double helixy composed by ATGC. Based on gene complementary policies, they composed with each other. In 1962, James Watson and Francisco Crick, together with Maurice, the name I kind of, Wilkins, Maurice Wilkins. Maurice Wilkinson awarded the Nobel Prize. How about Rosalind Franklin? Unfortunately, Rosalind Franklin had ovoid cancer. She passed away in 1958. If she's still there, the contribution would be more credits for her. Now, James Watson and Francisco Crick took Rosalind Franklin have a trip to the United States and around the world before she passed away as awarded for that. So that's a story. James Watson, when they have this published paper and have this great founding in the world, what they did? What they do in England? They go to the pub. They go to Eagle Pub. It's a bar, you know that. Have a beer. So they buy everybody a beer, a cup of beer. 2 pounds, 5 pounds. And they share the great news with everybody there, and nobody knows what happens. But they are happy, and the whole world is very happy. If you go Cambridge, if you have a trip, go to London, in the suburb of London, about like 1 hour from the downtown, in the eastern side, you go to the Eagle Pub, you will see James Watson. They have a template there, plaque there, which is talk about that. So this is a story which is about where in 1953 we find the DNA structure. Okay, so what is DNA is the question. So we want to talk briefly. What is DNA? 2'-deoxynuclear gases. What does the structure looks like? It is very simple. 5 carbon ribose. And this one, we usually go on the other side. So this is H, this is O. This is the base. So, what does the DNA looks like? Very simple. A base, A, T, G, C, a 5 carbon, 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. No O, deoxy. Nucleotides. So, as a DNA, there are only 3 major components. A base, a 5 carbon sugar, which is deoxy, ribose, and a phosphate. That's why I always say DNA is a very boring chemical compared to protein. Protein, you have a primary structure with amino acid sequencing, secondary structure like alpha sheet, alpha helix, beta sheet. By the way, who found the alpha helix? Linus Pauling. Did you learn in your chemistry class Pauling exclusion policy, which is arrangement of the electron outside of the chemicals? That's Linus Pauling, a great scientist. He did another contribution, nobody knows. He was awarded the Nobel Prize in 1970 because he sent a letter to President Eisenhower by the time to not using nuclear weapon in the world war, because that's going to be horrible damage for the whole world. So that's a great people. So we go back, talk about that. Deoxyribose, base, and phosphates. Okay. What are these? These are the base. What is A? Adenine. We got a purine. And the cytosine. This is we call purine. What is T? Thymine and guanine. Okay, it's called a pyramid. And how they combine together? A complementary with T, G complementary with C. Sorry. This should be guanine. This is cytosine. How they connected with each other? Hydrogen bonds. So that's why this is a relatively weak bond, and you could separate that. What is the structure of that? This is very simple. 6 carbon and 5 carbon. And pyrimidine only with 6 carbon. Okay, if you want to draw them, we could draw adenine very similar like this. NH2, adenine. How about guanine? Guanine and adenine basically are very similar. It's only a very slight difference. The only slight difference is this part is the same. This will be as a carbonyl group, and this moves to here. This is N, and this is N. So we go this guy, we go this way. Yeah. That's adenine, that's guanine. So what's a thymine and a cytosine looks like? Thymine, that's thymine. What's a cytosine looks like? Very similar. These are the same. You exchange this guy. Go here on the top NH2 and the same. Okay, so that's thymine. That is cytosine. How about the other guy? Do you remember uracil? Uracil is only in RNA, not in DNA. What's the difference? This becomes H. And we say carbon number 1, number 2, number 3, 4, 5, 6. This is in an unimportant place. Change the methyl group with a hydrogen group and change at an unimportant place. I just draw this for you. You don't have to remember. We don't test you, okay? Biochemistry teacher will ask you to draw. I think it's not difficult. That's a DNA structure. Okay, how the DNA gonna be connected? How DNA connected with each other? Five prime to 3 prime. How we do that? Okay, you have a base, H, 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 H. Actually, it is O, H, CH2, goes here, O, P. Okay, I'm going to draw another one right here. That's another base. Let's say T, H, 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 OH, H, CH2. So you have a O, you have a P. Are they connected with each other? Like here. This is 5 prime, and this is 3 prime. This is 5 prime, this is goes 3 prime. And what the bond here they call phosphor by ester bond. So always remember the DNA connected is from 5 prime to 3 prime. Okay? Let's see how many times we left. Yep, always from 5 prime to 3 prime. This is the slides. You could see there is some of the structure. Now, another thing we want to mention, when you have this single one, you have a base, deoxyribose, you have a phosphates. This part, we call it a nuclear tie. Connected with the phosphide, a nucleoside. Connected with phosphates, we call it a nuclear Hi. And then they're going through polymerization, have phosphodiester bonds connected with 5' to 3', going through polymerization, and we call this is nuclear gases. We just want to make sure this is nuclear gases. Okay, so that's what structure looks like. And this is slides which tells you A complementary with T, G complementary with C. A and T have 2 hydrogen bonds, G and C have 3 hydrogen bonds. This is important because that's the reason when we design a primer, we need to thinking about the GC components if it's in the research. This is another way which is very clearly to show you the glycosidic bonds between 3' to 5' and the diester bonds, and the rounding, just 2 strands rounding opposite direction. One is 5' to 3', another one 5' to 3' go the other way. Okay, always anti-parallel, and they are rounding opposite direction. You can see it referred as ribose sugar rounding the opposite direction. You see the sugar? One is go this way, the other one is go the other way. Okay? So that's a very interesting topic we covered about that. This is another way we talk about that. This is give you another picture, which is if you're not very familiar with the DNA structure and your background is not food, is not a chemistry or biology, we just give you another slide there which will let you easily to understand. This we will talk next week after spring break. You already know that DNA replication, we call it a semi-conservative replication. So they use, when they break down the double bonds, and they were using them as a template to biosynthesize their daughter bonds. And if there is no mutation happens, they should always be exactly the same. However, they always have something different happens. You can imagine you look like your parents, but you're not the same as your parents because there's a mutation happening all the time. Okay, so DNA replication is a detailed story behind it. We will talk about this after the spring break. So here we mentioned about the DNA basic structure, the history, and the semi-conservative replication gave you an introduction. Okay, so we start from here, the new material, and we're going to go ahead to talk about the case study. Oh, I didn't add something. Okay, never mind. I can. Yes.
The case study, there's no link.
Yes, sorry, I realized it. I will be re-added. Okay, I need to go back to see my other class. Right away a second. I said some accident happens, so let me go back to see my another class. I'll use the material to talk about that. Only several case studies. It says I will be. Wait a second. That's my another class where we'll talk about is practice exam 2, right? This is the one not going to test you guys. Exam 1. This is we already talked. Let me see this week. I forgot to. For some of the accidents, I didn't upload it there. Let me see. This is. Sorry. Okay. This is the case study of what I'm referring to. Okay. And I will be repost this after the class. So again, we're going to give you a list of the bacterial pathogen, and we'll give you a story, and then you pick from there what type of the pathogen it is. Most of these are very similar to what we talk about in the class already. So let's look at these questions. I will re-upload real quick after I go back to office. On June 22nd, 4 people who attended a cookout find razors for their local juice were hospitalized for 24 hours with difficult swallowing, double vision, and slurred speech. If you see these type of symptoms, what this means? This is a neurological symptoms. Many patrons of the event recall some level of mild stomach upset in the late evening of the event. It is significant to know that many of the dishes were prepared for local individuals from home canned foods. That is important information, from home canned foods. Laboratory lab find there are gram-positive blood with subterminal endospore, it's an endospore pathogen, and grow anaerobically, not aerobically, from a canned yellow tomato dish. What type of the pathogen is this? So, we talk about in the class, the gram-positive rods have endospore-forming bacteria is which? It is Clostridium. Is that right? We mentioned about the Clostridium. Then which Clostridium relate to the canned food? Yes, Clostridium botulism. So, the answer is very simple. It is Clostridium botulism. I remember we mentioned about botulism, we mentioned infant botulism, wood botulism, and food botulism. And the botulism here is home canned foods. That's extremely important information, okay? So we will, all these case studies, I hope you guys can print it out and took it in here. In the exams, it will have some very similar case, or maybe the same case. Okay, so that's important. Okay, let's look at the second question. On June 15th, a 70-year-old female was in her normal state of good health when she developed a fever for several days. She had no localized symptoms except for developed a large circular rash on her back. Well, that's important. A rash, circular rash on her back. Her history was notable that 3 weeks later, she had spent a weekend camping, biking on the trail in Mammoth Cave National Park. It is in Kentucky, Bowling Green, Kentucky, near Nashville. Okay. Her blood was cultured unsuccessfully, but presented with antibody titer against the typical spiral sheet disease. This is important, a spiral sheet. That is endemic for their region of the United States. The technician decided to view the blood sample under dark field microscope. What is the most probable etiological agent of this disease? It is a spiral sheet. It happens for when they go to the Mammoth National Park, it is a trail, and the symptom have a circular rash on the back. Which pathogen from the listed here? It is a typical symptom of Lyme disease. Yes. So which pathogen causes Lyme disease? Borrelia burgdorferi. And I mentioned that the burgdorferi is a doctor's name, which is Dr. Burgdorferi in Montana State University back in 1972. Lime County is a small county in Connecticut. Is that right? The target eye rash from the back is a typical symptom for Borrelia burgdorferi. And it is a very typical spiral sheets. And to identify spiral sheets, we need using dark field microscope. That's a very good question, to be honest. Okay. Okay, that's good. During the month of June 2010, the Kentucky Warren County Health Department identified 31 persons who had diarrhea with 10 days visiting of a beef cattle operation in Warren County. The patients ranged age 6 to 10. Symptoms reported 21 patients including blood diarrhea, fever, vomiting, and hemorrhagic uremic syndrome. Okay, I haven't talked about detail of this one yet. We'll talk later. All patients attended 3 elementary school, visited the farm. Approximately 300 persons visited the farm during the month of June, and stool specimens were plated on the McConkey agar. Find it's lactose positive, and there is a cell wall antigen comes out. Which pathogen causes hemorrhagic uremic syndrome from E. coli? I mentioned in the first class, but didn't mention very much detail. And you should know this. This is rarely what is this class for protect yourself. Never eat an undercooked beef steak. Which pathogen will cause? E. coli 0157H7. And you look at the news all the time. There is 50 pounds, 5,000 pounds of the beef by some company in Indiana. Then by some of the cattle operation in Colorado. E. coli 0157H7 will cause hemorrhagic uremic syndrome. It is a typical symptom. And since it's E. coli, it can grow on the McConkey agar and the lactose fermentation positive. Okay, that's a very good story for that. Okay, next one. A 60-year-old male was admitted to hospital. 5 days after prostate surgery, complaining with dysuria, which means difficult to pee. Okay, a terminology called dysuria. A quantitative urine culture was obtained, revealed E. coli is in significant number. Remember, E. coli usually will cause UTI. The physician placed the patient on a 7-day course of cephalexin, which is antimicrobials. Okay, remember, this person, this gentleman, is using 7 days intensively for antibiotics. The urinary tract infection improved, but the patient later experienced multi-water, loose stool cramping, abdominal pain, and dehydration. The physician ordered AB toxin on a stool specimen, which returns positive. The patient was diagnosed with pseudomembrane colitis and was unsuccessfully treated with typical antibiotic therapy. A fecal transplant was performed. It's horrible, I said, don't do that. This means a pathogen caused intensively antibiotics using. Finally, we'll have pseudomembrane colitis, and it is pretty severe. You have to do fecal transplant. What is the pathogen of this one? It is a Clostridium difficile. Acronym, we call it CADIF. Very good story for this one. It's a very typical story. Okay? Let's look at the next question. Bladder plates can be used growing anaerobic bacteria that cause gas gangrene. That's interesting. The organism was isolated from biobag anaerobic system and displayed a typical hemolytic with a disease looks like that. It's a double beta hemolytic zone. Okay. Which pathogen caused gas gangrene? It's Clostridium. What kind of Clostridium? We already talked about botulism. We already talked about difficile. You know Clostridium, tetanus. Then what is the other one left? Clostridium perfringens. Is that right? That's Clostridium perfringens. Has a double beta hemolytic zone on the blood agar. Okay. That's very important for this information. Okay. We will this sheet, this part of the case study sheet, I will be send it to you for you to study. Okay. Okay. Last one. In order to help you, I need to put another sheet there. So, because I do believe, if I don't do this, it's not gonna looking good. And you certainly don't want to retake it. That's not much meaning, wasting time. You have other classes needs to do. So, I'm maybe gonna put another study sheets there for you to do the practice. I'm gonna put this one there. Not done yet. Okay, I'm gonna put this one there for you to study. Good. Can grow on bioacetylene agar, hydrolyzed bioacetylene. What is that one? Enterococcus faecilis. That is a group D streptococcus. Okay. Which pathogen produce toxic shock syndrome? Toxic shock syndrome related to Staphylococcus. Yes, Staphylococcus aureus. Okay. Which pathogen may produce beta hemolysis in the enzyme coagulase? Which one causes coagulase? Staphylococcus aureus. Catalase test negative gram-positive coccell belonging to immunological group A susceptible to bacitracin. What's that? Which one is group A streptococcus? Streptococcus pyogenes. Okay. Alpha hemolytic gram-positive coccell kicks a catalase negative, sensitive to bile salts, and susceptible to opticane. What's that? Streptococcus pneumoniae. That's the first one we talk about. A gram-positive coccell that is catalase positive, coagulase negative, resistant to low dose of novel biosyn. Remember the story? How come the lady will know, the mom will know the girl has a problem? That's a honeymoon bacteria, Staphylococcus aureus. Okay? Can grow and ferment lactose on Microbial agar, that's E. coli, and also Klebsiella pneumoniae. This microorganism can grow the selective agar 7.5% sodium chloride. Does not coagulate the rapid plasma and susceptible to novel biosyn. Well, that's Streptococcus epidermis. It's a Staphylococcus epidermis. It's a normal bacteria. Okay. Belonging to a group of alpha hemolytic normally lives in the upper respiratory tract and may cause opportunistic infection. Which one is alpha hemolytic? Veridam streptococcus says. So veridam strep. Okay, I'll put the answers there. You can study by yourself. Gram-positive endospore forming bacteria in the soil. This one I didn't mention. It's bacillus. Okay, I will just mark it. Said I didn't mention. These gram-negative logs grow rapidly on blood and the chocolate agar, producing mucoid colony. Ferment lactose on Microbial agar. What's that? Klebsiella pneumoniae, there it is. Fastidious bacteria only grow on the BG agar and takes 5-7 days. Oh, this one you don't have to worry. 12 I didn't mention at all. What is the pathogen called whooping cough? Yeah. Very good. Gonoteropodiosis. Causing gram-positive logs forming palisades arrangement. The first one we talk about. Which one? Corynebacteria diphtheria. Gram-negative grow on chocolate agar, not on blood agar. It's a fastidious bacteria, causing meningitis and epiglottitis for age 2 to 5. What's that? Yes, HIV. Haemophilus influenza. I think you guys study very well. I mean, if you have some reaction there, that's really good. Bull neck. Pseudomembrane greenish. What's that at the throat? The first one we talk about. Corynebacterial diphtheria. HIV vaccine for the patient. Haemophilus influenza. What's DTaP protection for? Diphtheria, tetanus, and pertussis. Which bacteria is related to the belonging to Rebecca Lansfield group D? Yeah. Enterococcus facilis. Last one. Which pathogen will have rheumatic fever and glomerulonephritis? And I make a joke before I said this is an excuse you don't come to the class before COVID-19. Strep throat. Streptococcus pneumoniae. Okay? Good? Okay, so I will put that sheets on the eCampus. You guys go ahead to study, and also the case study, I will put it there. You can bring it. This will give you something to prepare for the exam too.